Let me introduce our speaker of the day, which is Dr. Chuan-Chin. Dr. Chiao studies visual neuroscience for more than 20 years. He mainly focuses on the retinal neurobiology, and he's interested in understanding the cognitive behavior of cephalopods. Right? He's been a faculty member in the National Tsing Hua University and now a Director General at the National Museum of Natural Science in Taiwan. Let's welcome him and his knowledge.
Okay, thanks, Sean. Very kind introduction. So in the next 30 minutes, I'm going to share with you some of my research in the past about 10 to 20 years study. So a lot of people ask me why I study cuttlefish or cephalopod in general. Or I would like to quote August Crow has said this words about organisms that exhibit extreme adaptation may reveal general principle not readily available in less extreme species. So while we are studying monkey, we study Drosophila, fishes, and other model animals. But this animal has unique feature. For example, as you see on this video, see the body pattern change rapidly. And those are unique feature which cannot be seen in other animals. So we call rapid neural polyphenism. So hopefully by studying this animal, we can learn a lot with other from other species. So for those of you who are not study animal, particularly invertebrates, let's just put everybody in the same page. So the cephalopod belong to the group called the phylum of mollusk. Mollusk means a soft body animal. And this group animal include lots of animals like snail on the land or clam, mussel in the sea. Those animals have shells. For example, but those only the modern cephalopod except the nautilus, which has a shell. Other modern cephalopod like octopus, squid, and cuttlefish, they are no shell. And because this modern cephalopod lack outer shell, and they must compete with fishes. And from the fossil record, you can see that these modern cephalopod appear around 350 million years ago, and away this appears Tilias fish, which is a fast-swimming vertebrates, and they are competed with same marine resources, so they must be very competitive in order to compete with fish. And in order to compete with this fast-moving vertebrates, if we look at the brain, and we can see that the snail, as I say, is one of the mollusks, a soft-body animal. They only have 10,000 neurons. The fruit fly we study in the lab is about 100,000. And the honeybee, our favorite animal, is about 1 million neurons. But octopus, of course, is a big, giant Pacific octopus, has more than 500 million neurons. This 500 million neuron is equivalent to cats and dogs, about 600 million. Of course, it's very far away from human, which probably have 100 billion neurons. But if we compare the brain-body ratio of cephalopod, you can see this diagonal line. On the left of this diagonal line is the bird and mammal, and on the right of this diagonal line is fish and reptiles. And those blue dots represent the modern cephalopod brain and the body ratio. So you can see that sitting in between birds, mammals, and fish and reptile. And if you look at the evolution from the tree of life, somewhere around 600 million years ago, vertebrate and invertebrate diverged. And in one end, we can see that vertebrate group, which have a very smart animal, intelligent animal, even consciousness animals such as bird and mammals. On the other hand, the mollusk, particularly cephalopod group, has independently evolved a very unique and a very complex nervous system. So if one would like to understand how human or how vertebrate evolved a highly conscious brain, maybe the natural control group is cephalopod, which is a cuttlefish I'm going to introduce you today. Okay. So I'm going to show you some of the results in our lab to showing that a unique feature, a unique cognitive function of this cephalopod, particularly cuttlefish. And first of all is the number sense. So we know that in human, in primate, and in some birds, we show that we have two independent systems for number discrimination or number sense. One is called subtilization, meaning if any number is equal or below 4, we don't need to count. We can immediately recognize the number. For example, if I ask you, well, how many colors in this random stars? You can say 4 colors. You don't need to count. You see 4 colors. But if I ask you how many stars in this cluster of stars, and if you count, you will know that it's 10 stars. So that's the counting and the subdivision are 2 independent and different numbering system, number discrimination system in primate. But whether in other animals also have number sense, that's the question we like to ask. So in human, we know that if you have number sense or number discrimination ability, that's very helpful. For example, if after this meeting go out for lunch, then you can know whether you give the money right or wrong. But for other animal, why animal have to have a number sense? There are many different functions people propose. For example, in fish, when they are trying to find a mating group, they will try to find the large group of mating groups. So it's a mating strategy. And for honeybee, they need to know where they visit the first flower, second flower, or third flower. So counting landmark is also important for honeybee. And in the mammals, for example, wolf and other dogs, they need to know which group are the large group. So for social interaction, of course, for the foreign behavior, for finding the food, also important for animals. So before we started this experiment about 6-7 years ago, we asked the question whether cuttlefish have number sense. So my student surveyed the literature and find that more than 30 animals, now it's about 40 or 50 animals, have widespread this number sense. But there's no literature or no evidence to suggest cuttlefish have number sense. And because, as I said, cuttlefish are a group of cephalopod animals, and they have a complex brain. So we thought it should be an easy experiment. We can demonstrate that they have a number sense. So that's the question at the beginning. We want to ask whether do cuttlefish have number sense, number discrimination ability. So in order to test this, we need to test whether the animal, the number sense is innate, it's not learned, because we know human, we don't need to learn which number are large group or which number sense, number discrimination. We inborn, we are innate have this ability. So we like to test whether cuttlefish also have innate number sense. In order to do so, we use the preying behavior, which is the natural behavior. So this is the video we show you is how the cuttlefish catch the shrimp. So they will immediately, when they find the shrimp, they will shoot out, it's called tentacle, to catch the shrimp very fast. And that's innate behavior, because we don't need to train animal. Once animal hatch from the egg, about 7 days, when they finish using the yolk for the nutrition, they will immediately start this behavior and without any learning. So we are going to use this preying behavior as a way to test whether they have a number sense. Okay, so the experiment is very simple. We use the people here probably familiar with called two alternative force choice design. Simply, we put one shrimp in one transparent plastic box, and the other maybe 2 shrimp in other transparent box, and there is an intruder sitting in between, and we create this dashed line, which is an imaginary line. So whenever the cuttlefish pass this imaginary line or shooting out the tentacle past this imaginary line, we know they make a decision either choose one shrimp side or the 2 shrimp side. And this experiment, we in order to make not a lateral or a side effect, so we alternate 2 shrimp, 1 shrimp every other times. But in this experiment, we only test 6 tests in one trial. We are not we did not test the animal beyond these 6 tests. It's because in the previous video, you saw that when we present the shrimp in front of cuttlefish will immediately grab the shrimp and eat it. And that's the native behavior. But in this setup, the animal cannot catch the shrimp because it's a plastic the box. And instead, they will shoot out the tentacle and punch on the box, the side, the wall. So they will get pain experience. And before, in about 60 years ago, in a Nature paper, people already showed that if you do the experiment many times, and one day, after 24 hours later, they will not do the experiment because they have this pain experience, pain memory, and lasts for at least 24 hours. So in order to not to do this 10 experiments in this our animal. So every time when animal shooting out the tentacle or the body move across this imaginary line, we immediately remove this apparatus so the animal will not touch or hit out the wall of this box in order to make sure that they will not generate this experience. But after 6 tests, we will leave out the apparatus and give the shrimp as a reward. But this reward is not train the animal to recognize one shrimp side or the other side. It's rather to make sure the animal will do the experiment again next day. So that's the trick. Okay, let's see the first trial. When we did the experiment the first time, we give a very simple test. It's 1 versus 5. Okay. So if you see this, we put the 2 chamber, 1 on the left and 5 on the right. And the cuttlefish immediately see that in the 5 shrimp side, there's a large number of quantity. So they will immediately go to the right, which is the number 5 animal. And of course, we can quantify whether they can distinguish. So we have test 12 animal, and each animal we test 18 times. So you can see immediately that cuttlefish prefer large quantity in this 1 versus 5 choice. And since we can do 1 versus 5, then we can do 1 versus 2, 2 versus 3, 3 versus 4, 4 versus 5. You can see that the even in the 4 versus 5, this very similar number, they can still distinguish, although the difference is smaller, but nevertheless, it's a significant difference. And we also quantify the reaction time, reaction latency around this different from easy to difficult test. As you can see that from 1 versus 2 and 4 versus 5, the reaction time actually increase. And this result lead us to say that, to conclude that cuttlefish number sense is not like human using subtitration, which is do not count. And the cuttlefish number sense is actually by counting rather than by subtitration. Okay. And in order to show you that the animal really has hesitation with difficult tasks, so I've shown you this video, which is animal when facing the 3 shrimp versus 4 shrimp. And you can see that when we put on the apparatus and the 4 on the left and 3 on the right, they see the animal sees 3 and they see 4 and they see 3, and finally got the number 4 shrimp side and then make a choice. So they also have to count how many shrimp in that box in order to decide which side has a large quantity. Okay. But in order to conclude that animal do count rather than using other cue, so we have to do 2 control experiments. First is density control. For example, the animal may see that the more dense box are the large number, so they may use density as a cue. So we try to avoid this, to exclude this effect by putting a transparent wall in the center so that we can separate the animal, make sure that both sides has equal density. And with that setup, we ran the experiment again, and you can see the result that animals still show a large quantity preference. So that without in the same density, it's not a density effect. It's really they count the animal, they count the shrimp. The other thing is maybe they are not based on the so-called density, but they are based on the motion, because the more shrimp side has more motion. So we have to make a motionless shrimp. In order to do that, we have a fresh dead shrimp, but because fresh dead shrimp will clump together, so we have to put in the line. So this is a front view. From the cuttlefish view, you can see that we put shrimp align this plate. So we make this motionless shrimp. Then we do the experiment again. And this experiment, you can see the trend is the same, but in the number of 4 shrimp versus 5 shrimp, they cannot distinguish without statistics. Although the trend is still similar, but they cannot distinguish statistically. That's because, I forgot to say, that's because in this experiment, we did not do 18 times. We only did 6 tests. The reason is because animals like live shrimp. And because we give them the dead shrimp, so they don't like dead shrimp, and we have to starve the animal for one day. And this is not good for animal welfare. So that's why we only did once. So only 6 tests rather than 18 tests. So the statistical power is not enough. That's why this did not show a significant difference between 4 and 5. And after we establish animal prefer large quantity rather than small quantity, then we say if we have 1 live shrimp but 2 dead shrimp, which is quantity is higher in the dead shrimp, and 1 is less quantity, but the live shrimp high quality, and where the animal choose. This experiment is very simple because before we did experiment, we already know the result. The result is, of course, they like quality more than quantity because they like live shrimp. So this result is very obvious. But the other experiment is very unexpected. For example, if we put the cuttlefish with a choice with 1 large shrimp and 2 small shrimp, both are live shrimp, and this quantity is equal. But if you, based on the previous experiment, we should hypothesize that the animal should choose the 2 small shrimp side more often than the 1 shrimp side, because this quantity, the number is higher than the 1 large shrimp side. However, the result is different because when we did experiment in the afternoon, we always got animal prefer 2 small shrimp. But in the morning, except the first one, animal always prefer 1 large shrimp. So they are morning and afternoon difference. And then we reason because animal in the morning, they are hungry because we didn't feed the animal all overnight. So they are hungry in the morning. So they choose 1 large shrimp when we did experiment in the morning. But in the afternoon, because after the experiment, we always feed them some small shrimp. So they are not so hungry. So in the afternoon, they prefer 2 small shrimp. And in order to make sure it's not a morning and afternoon effect, rather it's a metabolic effect. So we did experiment. My student comes to the lab early in the morning and fed the animal. So they make animal in the morning not so hungry, but the afternoon very hungry. And the result is reversed. So in the morning, if they are not very hungry, they show they prefer 2 small shrimp. Rather in the afternoon, they are hungry, they choose 1 large shrimp. In order to make sure this result is depend on the metabolics, that we need another control experiment. The first one is the size difference. And here we have 1 shrimp, 1 large shrimp, and 1 small shrimp. But the quantity is same, but the size are different. And we test this with in the morning and in the afternoon in the starved state and the satiated state. And the result is clear. No matter it's hungry or not hungry, once there is no conflict, so 1 large shrimp versus 1 small shrimp, they always choose large shrimp side. And the other experiment we have done before, but because we now have a small shrimp and large shrimp, so we did this again. So we have 2 small shrimp versus 1 small shrimp, 2 large shrimp versus 1 large shrimp, and both in the early morning, in the morning and in the afternoon, in the hungry state or not hungry state. And then we got the result that is also very clear. If there is no conflict, they always choose the large number side, no matter it's the 2 small shrimp or the 2 large shrimp. So from here, we can say that earlier human economic studies show that the human become more risk tolerant in their monetary decision as they get hungry. And so this finding is akin to the phenomena of economic decision making, whereby the risk attitude of human subject towards money, food, water, and reward change as a function of the internal metabolic state. So even though, as I said, the vertebrate, human and cephalopod are diverged 600 million years ago, but their decision making is still very similar in this respect. And in the previous experiment, I show you that the animal can distinguish integer number from 1 to 2, 2 to 3, 3 to 4, and 4 to 5. But can cuttlefish also distinguish fraction like 1.5 and 2.5? For example, 1.5 versus 1. And because we have small shrimp and large shrimp, if we consider small shrimp is 0.5 and the large shrimp is 1, then we can create 1 versus 1.5, like this case. But the most important, the key experiment, the way the animal can distinguish fraction is these 2 experiments in the red box, 1.5 versus 2, which means the 1 small shrimp plus 1 large shrimp versus 2 large shrimp, and also 2.5 versus 3, same thing. And the result is very clear that they can distinguish, particularly this one, 1.5 versus 2, they can distinguish significant difference. But the 2.5 versus 3, they cannot distinguish. And also we test the reaction time also show the increase, the trend of increase. That means also they are counting rather than subsidization. And this experiment and the previous experiment, if we consider altogether, it can be interpreted as they have the same just noticeable difference, or JND, we call that. People here in the audience, probably most people know the Weber fraction. Weber fraction means the difference between 2 numbers divided by the small number. And if we consider the first case, 1 versus 2, 2 versus 3, the absolute difference are 1, but the relative difference are different. 4.5, the relative difference or Weber fraction or just noticeable difference is 0.25. And if we compare this result with the fraction result, other result, 2.5, 2 and 2.5, the relative difference is 0.25, and 2.5 versus 3, the just noticeable difference or Weber fraction is 0.2. So we can say that from these two independent experiments, we know the cuttlefish number discrimination, J and D, is around 0.25 and 0.2. And something below 0.25 is probably difficult to discriminate. So in short summary for the first two experiments, this study demonstrated that the cuttlefish are capable of number discrimination, and the number sense is likely by counting rather than by subsidization. And their choice of prey number depend on the quality of the prey, because they like one shrimp more than 2 dead shrimp, and also appetite state, the metabolic state. And cuttlefish may equip this numeric ability of fraction, and if so, the Weber fraction is similar for both integer and fraction number discrimination. And if I have time, maybe 5 to 10 minutes, I'd like to tell you another 2 stories. One is whether cuttlefish can determine the prey value, because we know in the previous experiment, they always choose the large quantity or the large size of the shrimp. But if we consider this typical experiment, as I showed you before, that cuttlefish, if given 2 choice, 1 shrimp versus 2 shrimp, typically they always choose large shrimp size. But can we change the prey value of cuttlefish, and how do we change the prey value? The experiment is we present no shrimp versus 1 shrimp, because they will cuttlefish will always go to the 1 shrimp side. So after they go to the 1 shrimp side, we get a reward with 1 small shrimp, and we give them this reward for 6 times. So we call value establishment. We establish this now 1 shrimp is very good. So they change the 1 shrimp the value, and after that we immediately test this typical test of 1 shrimp versus 2 shrimp. As you can see, the preference change from they prefer, in this case, prefer 1 shrimp more than the 2 shrimp. So maybe we can say that they also know that less is more. I think that's a very philosophical, but at least in this experiment, we can interpret that the cuttlefish may know that less is more. Okay, besides less is more, we test how long this memory, this value change can last. So 1 hour later, we did experiment again. Also present the 1 shrimp, 2 shrimp. In this case, although looks like they still prefer 1 shrimp more than 2 shrimp, but there is no statistical difference. So at least we can say that they treat this 1 shrimp and 2 shrimp equally. Okay, and the control experiment is always important. So in this case, we feed the cuttlefish with 6 small shrimp, but without this 0 and 1 value change experiment. So in this case, they only get satiated. As I told you before, no matter it's hungry or not hungry, they always choose the large quantity. So this will also again test that animal without the value change, they always prefer 2 shrimp versus more than 1 shrimp, more the large quantity size. So suggest that the value change can be altered by this specific experiment. So the conclusion for this simple experiment is that the value of things is not absolute, rather it depends on context. So every time I give a general public lecture, I always show these 2 examples that people know what's the meaning of this experiment. The first example is a bottle of water may not have a high value in forest, but a bottle of water may be priceless in desert, so that's in context dependent. So the same bottle of water in different contexts could have a different value. And some people may not appreciate this kind of example. So I always give the second example. That's if your boss raised your salary by 20%, you would be happy. However, if you knew that your colleague was raised by 50%, then you will feel 20% raise is not very high. Of course, 20% raise is very high for everybody, but because we have comparative, we are relative value, so-called value rather than absolute value. So that makes you think that 20% is really depend on what other people get. Okay, So in the last 5 minutes also, I would like to show you some of the experiment that we are running, and may not show this emotion, but at least show that our attempt to answer whether cuttlefish have emotion. And most people know that in other animals, particularly invertebrate, I said this last Chica's lab in 2016 science paper showing using the judgment bias test to demonstrate that bumblebee could have emotion-like behavior. The judgment bias test is very simple. So they train the animal with different color. If the blue color discrimination, blue reward, but green without reward. And the test is not group red or not blue, not green, but rather in between, which is this red, blue-green color. And if the animal before the test give a small amount of juice, which is like give you a small amount of chocolate, which will make you very happy, then you will make optimistic decision, and then your decision time will be reaction will decrease. So in the sucrose condition, give you a juice, then a sucrose solution. In the intermediate, meaning the red, blue and green color, this ambiguous test, the pattern, the bumblebee will spend less time to make decision compared to without an expected reward. So that's the so-called JB test, the judgment bias test. So we use this judgment bias test to examine if cuttlefish have emotion-like behavior. So the control, of course, is without unexpected reward, always get the food. But the treatment is I randomly, sometimes we get reward, sometimes not get reward, which is a small shrimp. And to make this an ambiguous and difficult test, so we have three different tests. One is 1 versus 0.5, which is one large shrimp, one small shrimp. And small shrimp is half size of the one large shrimp. So this is easy choice. And 75% is the size is 75% of the large shrimp. That's a difficult test. And the equal size, of course, is ambiguous test. So in this case, we also test whether the reaction latency after the unexpected reward, treatment 1 is an expected reward, whether it will decrease their reaction latency. And you can see that in the this test is the difficult test and ambiguous test. They show the decrease of the reaction latency. So sounds like like they have this local emotion-like behavior. And we also test whether their decision outcome. So in this case, because it's 1 versus 0.5, easy test. So they are always choose the large size. And this is the number with the difficult test, but still with this treatment 1, they also show they prefer the large one, which is one large shrimp. But of course, in ambiguous, it's the same size, so they cannot distinguish. So it's 50-50 choice. However, the story turns. However, if we give expected reward rather than expected reward, we also show that the animal's reaction latency decreases. And this, of course, does not suggest they have an emotion-like behavior because it's not consistent with judgment bias test. So simply increase the appetite of cuttlefish would speed up the choice. And finally, another thing is also rule out this so-called emotion-like behavior is even without eating, without feeding the cuttlefish, even we just make them to go through this tentacle strike, which is preying behavior without eating, the animal still showing a decrease the reaction latency in the treatment if this group. So simply said, increase the internal drive of preying behavior will speed up the choice. But if we look at the outcome, there's some difference. Fortunately, there's some difference. So in this case, treatment 1, they can always choose in the right choice. But in the treatment 2 and treatment 3, which is appetite increase and the preying drive increase, they show decrease their outcome, which means they cannot distinguish 1 versus 0.5 and 1 versus 0.75. In this case, so they show a speed accuracy trade-off. But this is not the case for the treatment 1, which is an expected reward. So at the end, although this experiment does not demonstrate that cuttlefish have emotion-like behavior, but it does show that cuttlefish foraging strategy are influenced by the previous surprise event and their internal state. So with that, I would like to acknowledge all these students help us to finish the experiment, particularly when this fraction number experiment was done by a high school student. Thanks very much.
Actually, I'm going to take this chance to ask the first one.
Okay.
So humans, we have something we call ensemble perception.
Yeah.
Which is like a gist perception. Like coming to the room, we can kind of roughly know how many people there are without counting it. So do you think if you give these cuttlefish kind of uncountable numbers, large quantity of numbers, they will have something like that?
Yeah, I think so. But I haven't done experiment with large number. But I believe animal like cuttlefish have this ensemble cognition. Yeah.
Okay, and thanks for a very interesting talk. And I have a similar question to what Shao just mentioned. Even though you have controlled the density and the movement of the shrimp, is there also a possibility that the cuttlefish actually estimate the total volume of the shrimp in the box instead of actually counting it?
Yeah, that's very difficult experiment. We have been asked by reviewer for the same question. But of course, we need to do more control experiment, but that's really difficult to exclude because we need a specific experiment design to keep the quantity or the volume the same. But this is very difficult because these animals are freely moving, and the animal in the box, 2 box shrimp, also are real shrimp rather than simulated on the screen. So it's the control of the prey are very difficult. So that's a good suggestion, but difficult to test.
Okay, thank you.
Okay.
Thank you. Yeah, this is a very interesting research, and yeah, this is just curiosity questions. Do you observe that the cuttlefish have the sense of regular and irregular pattern, like when they face the prey with the same amount, and then the next time they will face the prey with different amount. Will it affect to the how fast they will catch the prey? So yeah, what I asked, do they have like a confusion when they see something new?
I think in that particular case you mentioned, probably they will show similar response because the animal probably not recognize individual shrimp. So if you present the 2 shrimp and 1 shrimp, they will prefer large quantity. But I can extend your question to whether they have an individual difference, and every time they will have different result. In other experiment, not in the experiment I mentioned today, we also, because in this experiment we don't need to train animal, but in other experiment we need to train animal to recognize specific pattern and reward this pattern so that we can associate the pattern with certain response. And in that experiment, we found that some animals are easy trained. Sometimes there are 25 trial, you can train animal, you can finish training, but some animals require 125. So that's, I don't want to call a smart cuttlefish and stupid cuttlefish, but indeed the animal showing different learning ability. And also the other experiment, we also show that animal response could be different. If you put the shrimp in front of the cuttlefish, some cuttlefish may use this strategy to catch the shrimp, and the other animal may use a different strategy. So they show an individual difference. And every time I talk about this individual difference, I always told the general public that most people think that it's bad for statistics because it's nuisance, because you run statistics, you don't like individual difference. But to me, it suggests that the cuttlefish are not something very rigid. They are flexible. They can have a different choice, and sometimes they are very flexible. And the reason we saw that the individual difference in certain circumstances, meaning that it's something we are not control very well. So that suggests that animals are very, I shouldn't say smart, but at least they are conscious about this environment so that they make decision based on a certain factor rather than the factor we design for. So that's why every time I see individual difference, actually I'm very happy because I can say animal is more smarter than your animals, something like that. Yeah.
Take one final question.
Thank you for the wonderful talk. I'm quite surprised about the result when you show they choose one shrimp, then there are the following small one. And so to me, so they keep this action policy. So which kind of imply it's model-free reinforcement learning, but so they reinforce this behavior without the internal model. But based on my understanding, many cuttlefish behaviors, they kind of show model-based way to plan their action. And so how do you think about it? Why in this situation they have a more model-free behavior, and then this other scenario maybe more model-based?
I probably cannot directly answer your model-free question, but at least I think animals know what's their choice. And this simple interpretation of our result is the experience dependent, meaning they are learned the one shrimp is higher than other. So we know that animals have, when they train the one shrimp has a reward, they always go with the reward. So even next time we have one shrimp versus two shrimp, the animal will go one shrimp. But they require other experiments to test whether it's a different situation, because if we give them not 1 versus 2, rather 1 versus 3 or 2 versus 3, and whether they still choose the smaller number rather than smaller number rather than large number, that's something we haven't done. So it's required further test to see whether animal can really derive this concept of smaller, less is more, always using this less and more principle to other tests. That we haven't done, so still more experiment to be done. Yeah, thank you.
Let's thank Dr. Chiao, and it's very interesting topic.
Thank you.
She was supervised by Dr. Cerebral and Tononi at the Center for Sleep and Consciousness at the University of Wisconsin-Madison. Yeah, so if this is ready, let's welcome her and her talk.
So today I'm going to introduce part of my thesis project titled Investigating the Role of Cerebral Cortex in Consciousness. So it is of particular interest for the basic science to develop a consciousness meter so as to ultimately inform us the conscious level of a given system. And it is also crucial for the clinical use, because for the numerous unfortunate patients lying on their bedside and able to communicate with the world, it is crucial that we know whether our loved ones are still there or not, because up to 40% of them actually are still conscious, but is unable to talk to their caretaker or their family. So in order to make decisions of whether we should continue the life-sustaining therapies for them, and whether they can still hear us or feel anything, we need to have a meter as such. Actually, we have a best tool to date, which is the so-called PCI or PCIST, standing for the Perturbational Complexity Index State Transitions. It is a mouthful, but all we need to know for the moment is that it is a consciousness meter based on the cortical evoked potentials, and that it reliably reflects the level of consciousness in humans without depending on report or response from them. So in this set of seminal studies, we see that on the x-axis is the conditions of physiological states, and also clinical anesthesia or pathological state. On the y-axis is the PCI value. So, for example, we see that in the healthy human subject, when somebody is normally awake, they tell you that I am conscious right now, I'm experiencing this or that, and that the PCI value is pretty high. And for people who are also normally healthy, but then we'll wake them up from sleep, and when they tell you that I was coming out of nowhere, I was experiencing nothing, we see that the value drops to pretty low. And it is also the case when we look at the slow wave anesthesia, and that in this whole population of 150 healthy human subjects, researchers can actually draw a line to tear apart the conditions where the subject tell you that unconscious versus the one that they're unconscious. And that in the end, we can achieve 100% sensitivity and specificity on a single subject level to tell you whether the person is conscious or not. So these are all great. And then this cutoff can even be so powerful that it can be applied to very tricky clinical conditions. So in the same graph on the left, we see that when the PCI is applied to what was carefully diagnosed as the vegetative state patients, the value is below the cutoff line, indicating that it's indeed unconscious. Whereas on the other extreme, on the very far right, we see that for the patients in the locked-in syndromes, meaning that those are the ones who are fully conscious, but just very unfortunate, and as if they're locked inside their own body, the PCI also detect that and show that it has a high value. So why does it work so well? It is especially in the way that it's based on the notion that for a system to be conscious, it needs to be both highly integrated and differentiated. And that different than other measurement of consciousness who look at the spontaneous activities of the brain, here we look at the evoked cortical potentials. So in practice, we firstly deliver a brief perturbations, and classically it's going to be TMS, a magnetic pulse, and then measure, and classically with EEGs for the electrical signals across the cortex. And once we obtain these trial and repeat it for hundreds of times, we align those trials together upon the time of stimulations, and we compute the spatial temporal complexity of those response, and that gives us a final value in number of the PCI. So what I'm here to address, this is going to be a short and sweet story that I tell you, and I know that I'm one way in between of you and lunch, so yeah, please, it's going to be short. So hopefully very interesting. So the first question that I'm interested in address is that I wonder what happens if we apply that to animals for a tool that is so powerfully working in human. So because to me, I infer that animals are conscious, in principle not different from the way that I infer that my fellow human friends are conscious. And that on the other hand, for a measurement of consciousness to be fundamentally correct, ideally it should account for not only human consciousness, but also the consciousness of other things. So I want to firstly see if it works in mammals, say rat and mouse at all. And second, if it does, it's actually going to be great for us to use it as a model to study the neuromechanisms underlying this PCI. So because although the PCI works so well, it is poorly understood for its cellular and network mechanisms. A long-standing hypothesis, though, is that for the state where we see the PCI is low, there are actually states that are with bistabilities, meaning that there are slow wave EEGs and that there are cortical neurons firing more or less synchronously altogether, and then more or less all silence and transit between these periods. So when we apply a perturbation in a state like that, the system actually necessarily fall into an off period of neuronal silencing. And it might be that silence that break down the causal interactions of the system, leading to a low PCI. So lastly, I want to test, because so far, the way the PCI is computed is that this responses that's obtained across the whole cortex. But I'm curious to see whether it is enough actually for us to have a reliable PCI just by looking at the responses from single cortical areas. So with that, we have the study that is published earlier this year with Dr. Matthias Cavelli and our team. And I'm going to firstly show you the results that we got from rats and then mice. Okay, so because we want to firstly test whether the PCI changed across states in the same way that we see in humans. So we want to firstly implant the rat chronically with electrodes to give the perturbation and also the electrodes to give the measurement, which compute the final PCI. So to do that, we chronically implant the rat with the neural nexus probe on one side of the brain, and in this case, it is the left side of the posterior part associated with the cortex. We also have all the different other combinations just to see if there is any regionality at all. And then on the other side, we implant the cutting-edge neural pixel probes with 384 electrodes and recording the neural response in a single area, but across the layers of the cortex in the front and back, for example. And that once the rat recover from this state, and we start to do the recordings in the freely moving, so continuously for 24 hours every day. And that, for example, in the graph on the bottom, we will see that it is showing you the 12-hour recordings of the brain signals, and here is just presenting the amplitude of slow wave activities, which can mark the state, a physiological state of the rat. So here we see that across the state of wakefulness in white and non-REM sleep, REM sleep in different colors, different stages of the sleep, we deliver the perturbation, the electrical pulses, and that with a final goal of collecting around 100 trials per state. So once we get this data, we can start to compute the PCI. And we do it in the way to firstly pull together all the trials that we obtain from each state of each recording site. So here is one example that during wakefulness and recorded from the posterior parietal association cortex. And after averaging all those trials, we have a good flat baseline. And then upon stimulation, there is a pretty complex looking response that lasts for about 1 second. And that there is also differentiation across the electrodes, meaning that the neurons of different layers, for example, are doing different things at a given time. And how do we quantify the complexity of this response? So first, we perform a dimensionality reduction to get the principal components that index the spatial complexity of this response. And then for each principal component, we do a recurrent analysis that in short will give us the temporal complexity of this response, so that in the end, once we add them all up, we'll have the final PCI value, which is the spatial temporal complexity of the whole ERPs. And in this case, the value is 25. But what does this number mean? And then we go to the next slide. And can see that it becomes meaningful once we compare it with other states. So in non-REM state, we see that the value is pretty low, whereas in REM sleep it's up high again. And this is actually also what we see in human, which is very exciting. So we know that REM sleep is what's historically known as the dream sleep. And indeed, almost always when you wake somebody up from REM sleep, rapid eye movement sleep, he or she will tell you that I was just dreaming of experiencing this or that. And whereas non-REM sleep, the non-rapid eye movement sleep, is the ones where more likely when you wake somebody up from that sleep stage, he or she will tell you that I was not experiencing anything. So this example is very consistent with what we see in human, and that across the population of rats, this is also what we see. So on the bottom left is a cohort result from other rats presented as a percentage of wakefulness results, so that we see this V-shape, meaning that the value is consistently high in wake and REM and low in non-REM sleep. And that this is true regardless of the recording or stimulated site. And that on the bottom right, we also see that in the additional anesthesia condition, the value is also very low compared to the wake, as we see in human. So far, I've demonstrated that the PCI also work in in rats, like what we see in human, and that we do not actually need to obtain a signal across the whole cortex, but it can actually be achieved from single areas. So with that, we can then look into the mechanisms and see if there is indeed a off period associated with the low PCI at all. And here what we see is that on the top is what we're familiar with, the ERPs, and that in the stage of non-REM sleep where the PCI is low, in the middle, I'm showing the raster plot of firing, in which each single dot is a single unit, and that each single line is a trial, so that upon stimulation, there is a big blank following that, meaning that there is indeed some neuronal silencing going on. And to better quantify that, on the bottom we're seeing in histograms, quantifying the number of the single units that we see in the middle, so that once we set a threshold, we see the duration of the off period is like 30 milliseconds during non-REM, but is non-existence in the other state, supporting the hypothesis that it may indeed be this presence of off period in the state of low PCIST that leads to a breakdown of the causal interaction. Okay, so briefly, I will go through the result from the mice. So mice is a species that is closely related to rat, but it's much smaller and has a less developed cognitive capacities, but it can still interact with you in a very complicated way with every very temper, and that it can also perform very fairly complicated behavior tasks like pressing lever, like licking spout, etc. And here it is very powerful actually to use mouse, also because it has a huge library of transgenic mouse line, and that it can empower us to use the optogenetic manipulation much easier. So here I'm curious to see for the first time what happens if I perturb the system not with the classic TMS or electrical stimulations to compute the PCI, but rather for the first time using the optogenetic stimulations. So here just briefly for this technique, I express this traneurodopsin, which is the excitatory member protein in the neural population of my favorite. So here I try to target the pyramidal cells of the cortex across layers and across areas. And I achieve it by crossing these two transgenic mouse lines that gives me the offspring, which express this opsin very nicely. So if we click once more, we can see that it is expressing beautifully across layers in different areas of the cortex actually. So here is a chrono slice of the mouse brain, and that is lighted up in green because the opsin, each one of them is linked with the green fluorophore. So we could go to the next slide and see that with the same line of thought as what I just introduced for the rats, here I do it for mice, and that instead of implanting electrodes to deliver the electrical pulses, I implant the optic fiber to give laser to the instrument, and with different combination of stimulation and recording side. And that once I do experiment, okay, and see the result. So here, with the result from the mouse posterior cortex, we see that the value of PCI is also high in wake and low in non-REM anesthesia, like what we saw in rats and actually also in humans. And that in terms of mechanisms, in the next slide, the state with low PCI is also with the presence of off period, supporting the hypothesis. So far it seems that in the mice, it also works, and that the for the result with the electrostimulations, for example, which recruit not only cortical specific neurons. So for optogenetic stimulation, I should say that it is very elegant because it excites only the green neurons that we just saw. So it's the cortical pyramidal cells that I'm stimulating to better investigate the cortical mechanisms. Whereas for the electrostimulations, it recruits not only the local cortical neurons, but also largely the transpassing fibers. But here we see that what we saw with the electrostimulations or magnetic stimulations for that sake can also be largely explained by direct cortical stimulation. So in the next slide, I would wrap up and conclude that the PCI also works in rodents, like what you saw in human, and that it can be obtained from just single cortical areas. And then lastly, for the state with low PCIST, it may indeed be associated with the off period of neurosilencing that break down the causal interactions of the system, leading to a low PCI. So with that, I want to thank my amazing teammates and lab mates and friends. And in the next slide, I will thank you all for listening, and I'm happy to take questions.
Thank you for the great talk. So in the end, I think in humans you show the the EEG complexity, the PCI from both front, anterior and posterior recording? And in the mice, do you also have data from the anterior cortex? You seem to have only showed the posterior, right? Is that right?
Yeah. So that is the first catch, and I did that so as to, we can actually, instead of going back, I'll go forward. So like if we click for until the last slide, I think it is waiting for you. But I try to be very conservative and not claim much, but if we go even more, like passing the acknowledgements, more forward.
Oh, hidden slide? Oh, okay.
Yeah, so we see that it's not working so well in the front. It's inconsistent across the state in the mice, anterior cortex. So it is an open question about why we get what we get. So we say like different, ideas. So one possibility could be that say the mouse is just not exactly the same as rat or human. That is frontal cortex, prefrontal cortex is less developed. And it could also be that here I'm using optodynamic stimulation, which is so normal, and doing cortex, cortical neuron specific perturbations. And that could account for why I'm seeing not a big meaningful response when I record from a single area while stimulate the front. While record from the front. And then lastly, it can also be a matter of the front versus back event. That some part of the brain in some species, when it's organized in certain ways with the anatomy, it may not be the best candidate to sustain the causal interaction system thus consciousness.
So due to the time, I have to close this session. Let's thank all speakers again.